Today, Dr. Solomon Lane is here from the Claremont Colleges, and she'll give a talk entitled Growing Up in a Dynamic Social World, Early Life Effects on Behavior and Neuroendocrine Function. And we want to give her as much time to give her talk as possible, so I will not introduce her, but instead refer you to the link on her website where she praises us all so much more eloquently than I could. Please join me in welcoming our speaker.
I'm absolutely thrilled to be here. One technical question. Hopefully this doesn't distract you too much. I now teach hybrid completely, and I kind of like the subtitles. It shouldn't eavesdrop on you too much if I'm speaking, and then when we go to discussion, I'll shut it off, because then it definitely goes into eavesdrop mode. So thank you so much for having me. My name is Tessa. Like Susan said, I'm at the Claremont Colleges. I use she, her pronouns, and please, Tessa is fine for when we're speaking together today. So essentially what my research aims to do is to take baby animals and try to understand who they're going to become and how. And so I'll introduce you to these adorable little creatures in a moment. This is Burton's mouth brooder, a species of highly social cichlid. But this set of questions is not at all unique to this animal, right? Pretty much any animal you can think of, you can ask questions about who that animal will become in a variety of capacities. I like this slide, one, because, you know, why not start a talk with pictures of baby animals? Let me move the zoom. Okay, so now I can see the chat. But also to demonstrate that this is a completely universal question. And so no matter what your area of focus is, there's probably some element of this question in the work that you do. Especially when we're thinking about humans, all kinds of disciplines ask about who humans are, especially when they're young, and then who they go to become. And the perspective that I'm going to bring today is through behavioral neuroendocrinology. Though I have to say I first became interested in these questions about development, I think pretty honestly. Both my moms were preschool teachers, and I don't know if anyone in here has parents who were teachers, but when you do, you spend a lot of time not only in your school, but in their schools as well. And so I also have a little sister, but seeing people grow up is really just a phenomenal thing. And I came to this sort of observation, I think pretty honestly, but it's obviously an observation that pretty much everyone, I think at some point in their life, comes to both independently and through their studies, is the fact that seemingly similar individuals, even in similar environments, can behave very differently. And this is sort of where my interest launched from. First thinking about tiny humans in an educational setting, and then as I grew in my own education, really approaching this through the study of different animal research so that I could be more manipulative and access the brain in a way that just isn't feasible when you're talking about humans. And so for my work, I'm really aiming to understand social phenotype in the context of life history trajectory. So thinking about things like how individuals grow over time, when and how they mature, and the timing and the kinds of reproductive decisions they make within the context of their own social phenotype and also the social environment that they're in. They're still on the title slide. Okay, sorry about that. And so I'm going to first frame this in terms of developmental plasticity, which we can really think about as gene by environment interactions also interacting with development. And it's a process that allows individuals to alter their phenotypic development in response to both current and anticipated environments. And what's really interesting is that early life across species, humans included, it really has a disproportionately strong and long-lasting influence. Compared to experiences that happen at other times of the lifespan. And so focusing on early life really makes a lot of sense. There are a ton of examples to think about. I thought I would bring up one of the classic literature examples of phenotypic development, where we have these Daphnia, these tiny little pond creatures that if they develop in the presence of predators, they grow this really sort of remarkable armor on their head. And if they grow up in an environment without predators, they don't develop that. And I'm going to talk about a number of other examples in a moment. And so really what we're thinking about over time is this integration of many different genes, multiple important environmental factors, influencing at different times of life. And of course, an influence that happens early in life is going to affect phenotype, and it's going to affect the phenotype that actually encounters the next environment. And so all of this sort of accrues and builds on one another. And so in terms of a model, it becomes quite complex, but I'm going to talk about some of the experimental approaches to thinking about this. And I'm especially interested in early life social environments. Those are frequently made up of parent-offspring interactions. Early on in this literature, it really focused on maternal interactions, but I think it's expanding a lot into paternal interactions as well, in addition to sibling interactions. And I particularly put this kitten picture up here because that, I think, best encapsulates my relationship with my sister. I'm not sure exactly which one I am, but very loving. But, you know, a good pounce moment happens as well. And the reason we care about this is because those kinds of interactions, especially early in life, have all of these consequences. For example, for growth, activity and locomotion, things like anxiety and fear, social behavior like aggression, parenting, and play, social competence, so being able to match the social behaviors that you're expressing to the context that you're in, human physical and mental health, and stress axis function, which is going to be a big part of what I talk about today. And of course, one of the reasons we care about this is that it has consequences. So behaving differently and how your behavior matches your environment really does matter, and we can measure that in terms of fitness across a variety of species. I chose these, but there are many, many more. So for example, in these olive baboons, thinking about social integration, having a positive impact on birth rates and survival. Oh, apologies, this is the example for horses. And for humans, thinking about the strength of a social network having consequences for things like decreased incidence of mental illness, reduced severity of disease symptoms when someone is ill, and you're actually more likely to adhere to a medical treatment regimen if you have that strong support network. So we can measure this in a variety of ways across a variety of species. And for me, I'm currently studying this in Burton's mouthbrooder, which is a highly social African cichlid fish. Here we have sort of one of the brightly colored territorial males, an adult female, and I'm going to be talking a lot about the juveniles. They come from Lake Tanganyika, which is one of the East African Great Lakes, and the population that I have in my lab is descended from a wild-caught population, but it has been in the lab for many, many generations, so it is an actual lab strain at this stage. And one of the reasons this species was so appealing when it came to studying social behavior and later social behavior in brain is because of the kind of social communities that they form. And they show these really evolutionarily conserved social behaviors like aggression, affiliation, courtship, parenting, cooperation, not just in the field, but also when you bring them back into the lab. And so we have some validity there, even though, again, I will tell you this is an actual lab strain. So adult communities are made up of dominant males. In the lab, we use these broken terracotta pots as their territories. So students in my lab get very good at breaking pots with hammers in exactly the same way so that our environments can be as identical as possible. And these dominant males are brightly colored, they're territorial, they're reproductively active. And this is in contrast to subordinate males who are dull in coloration, they're reproductively suppressed, and they don't show aggressive behavior. They actually shoal along with the females who have a ton of cool behaviors that I'll talk about in a minute. And again, one of the reasons the species was so appealing is because not only do they have these distinct phenotypes, but the phenotypes are flexible. So a dominant male can transition in his lifetime to becoming a subordinate male, and in that transition, he will lose that coloration, his brain will change, his hormones will change, his testes will shrink, and just a completely different phenotype, but the same individual. And they will expect to change status multiple times over their life. Again, subordinate males can then ascend in status and become dominant, so they will gain that coloration. They will grow their reproductive axis. There are many changes in the brain and across their hormones as well. And so the fact that in the lab you can socially engineer this kind of dramatic transformation is really useful. And it can be used to the point of if you have a smaller male, let's say in a middle compartment of a tank, and dominant males on the side and a female available to him, he might flash this dark eye bar for the female and then sort of turn it off when he's near the other larger dominant males to not be as much of a threat. Okay, so I want to show you a little bit what they look like interacting. In this tank, there are about 8 males and 8 females. The males are brightly colored. They're either blue or yellow, and they also have these little bead tags on them. There are territories on the bottom, and basically what you're going to see is that these dominant males are total busybodies. So they're chasing other animals, they're having border displays with each other, basically sort of lining out in the tank where one territory ends and the other begins. And the subordinate males and females hang out up here in a shoal. So they're quite charismatic. Now, I want to show you another tank. This was from the same experiment that Isaac Miller-Cruz ran. Same number of males, same number of females, same tank setup, and one of the first things you'll notice is that where are the fish? So here in this tank, one of the dominant males has taken his territory to be basically the entire tank, and he is corralling everyone else into the back corner. So, right, so many questions. Why is he doing that? Why are they letting him? What are the consequences of this? But the reason I really like showing this contrast is that not only does individual behavior change, but it has this ripple effect for the entire social group, right? If you were to take him out, the majority of the social group would be the same, but it would be a completely different dynamic potentially, right? And all of this can be engineered by just adding and subtracting fish from the tank. Another reason this species is really great to work with is at this point we have really cutting edge tools to work with the brain as well. And so we can think about the neural regulation of behavior for multiple interacting fish in a social group, which is really useful. We have important areas of the brain identified and sort of what receptors are expressed there. And I'll say more about this as we move forward, but also where those brain regions compare across other vertebrate lineages. So this is a really sort of phenomenal foundation to be building these studies on. Okay. So for my work, thinking about what are the developmental origins of social phenotype. And so I want to talk a little bit about some of the experimental approaches. So if you're thinking about how individuals vary, and here in this phenotypic space, I'm showing animals that are similar as close together, and the ones that are far apart as more different. One of the things we often sort of hope and maybe have a myth around is that if we have variation in behavior, you're going to see that exact variation mapped in brain. But the truth is it doesn't map that cleanly. You can have instances where you have similar brain phenotype mapping to disparate phenotypic space, and vice versa as well, similar phenotype mapping to different brains. And there are different reasons for that we can talk about. Some is the fact that just there is no one way to measure brain, right? You can be thinking about neuron activity or cytoarchitecture, so how the cells are in the brain. You could be thinking about where receptors are that are interesting. I'm going to be talking today about cortisol and testosterone a little bit. Or you could think about where genes are being expressed. So there also just is no one single brain phenotype. There are also other layers between brain and behavior. And hormones are a really big one that interact reciprocally both with behavior and with brain. And so in my work, I do try to integrate across levels, but it poses challenges. And then, of course, the focus of my talk today is the fact that animals grow over time and they change. And so they can change in different ways. And in this field, one of the things we really know very little about is what's happening with the juvenile brain, how that develops over time, and how that relates to juvenile phenotype. So what I'm going to talk to you first about today is how early life experience is affecting the development of both brain and behavior. Second, thinking about what juveniles are actually doing in their social groups. 3, thinking about the neural regulation of juvenile behavior. And then hopefully, if we get to it, thinking about the behavioral and neuroendocrine regulation of status in both juveniles and adults. So the first couple studies that I'm going to talk about, I did as a postdoc at UT Austin in Hans Hofmann's lab. And this first study really sort of set the foundation for the work that became the focus of my lab. And so here, thinking about what's the effect of early life social environment on behavior and brain. And Pamela, Savannah, and Naja were really important to this work. And our hypothesis here is that rearing juveniles in different social environments, here I'm going to show you animals raised in groups versus pairs, would have an effect on multiple kinds of behavior, including social investigation, dominance behavior, and subordinate behavior. And so in this study, we took brand new baby fish. We put them directly into social groups, either of 16 fish or of 2 fish in a pair. And we did this because these different social environments vary in terms of the social experience that these animals will accrue and the social sensory cues that will be available. And presumably, the social groups are more complex. They'll have more total social interactions. There's a possibility for 3-way or more interactions. These fish can learn a lot by watching, just like we can, and so there are opportunities for that in a group. And they can experience different social roles. So you might be subordinate to one, more equal with another, or dominant to another. And so we kept these animals in these groups for 8 to 10 weeks. And during that time, I want to just tell you a little bit about the developmental steps that they're going to go through. So when these animals reproduce, they're called Burton's mouth brooder because the female actually broods the babies in her mouth. So she'll lay the eggs in the male's territory, scoop them up into her mouth where he fertilizes them, and they will go through the entire the entire larval development inside of the mother's mouth, packed in there with their siblings. So this is actually the very first social environment that they're in. And if you see here, these are little baby eyes sticking out from the mom's mouth, and this is my finger, so that gives you some sense of scale. And I'm going to show you what they look like. If you just gently squeeze her opercula, the babies will spin out. And again, that's my hand for scale, and this is her little nose sticking out. When the babies first are out in the world, they sort of shoal together. So this is just video taken when I directly it's called stripping the female, and put them, the babies together in a bucket. And they'll shoal for about the first couple weeks. And then they'll move into overt social interactions. Chasing, nipping, carouseling is kind of just what it sounds like. They're spinning around each other. I'll show you that in a minute. Tail beating, sometimes they'll hit each other with their tails. Fin spreading to make themselves look bigger. Sorry, this video is a little bit small. There's one baby right here and another baby right here, and you're going to see some really great chasing here in a moment. So starting to move closer to one another. See a carousel and some nipping. And then one clearly becomes sort of the aggressor. And they approach, and the other one swims away. They can get territorial pretty early, around 5 weeks old, and sort of take on a lot of the behaviors that the adults are doing. You can, in these fish, decouple chronological age from developmental age, so you can accelerate them or slow them down. So this would be thinking about sort of an accelerated time frame. And if they're going sort of as fast as they can first reproduce around 3 months old. That's not typical, though. So that's some of what they're doing during this 8 to 10 week time period. And then we measured behavior in 4 different contexts. We first looked at open field. Oh, I thought this was going to animate. Apologies. Open field is just the focal animal moving around the space by itself. We then added a small cue fish in a little vial on the other side from the territory to look at social motivation. We then added a smaller fish to basically ask the focal animal, what do you do if you're in a position to be dominant? And then we asked the focal animal, okay, if you're with a larger animal, what do you do if you're given the opportunity to be subordinate? Because really these animals need to be able to do both. We measured approaches, so one animal swimming directly towards another, and displacements or submissions, that approached animal swimming away. So we used principal components analysis to reduce the dimensionality of these data. We first found that the open field behavior, social cue investigation, and dominance behavior all differed based on early life experience. So the data I'm going to show you are PCA for each of these individually and looking at how they're interacting together. And so what we found, we've got groups on the left, pairs on the right for PC1 for open field, social cue, and the dominance behavior assay. And what we found is that animals in the groups were more active in the open field, more active in the social cue investigation, and more socially interactive in this dominance behavior assay. And what's really interesting is that if you line those up, they actually all co-correlate. And so what we see is that there's sort of one axis of behavior here where the paired animals tend to be restricted towards this lower end, and the group animals actually are represented the entire way along this continuum. And so what this means is that juveniles, again, that were more active in the open field, were also more active in the social cue investigation and more interactive as a dominant fish. We also found an effect on subordinate behavior. And so what we saw is there was a trend for pair-reared animals to have higher agonistic efficiency, for the large fish to have higher agonistic efficiency, meaning that when they're approached, they're really likely to swim away. So they're not going to stay there. And they have a lower efficiency asymmetry. So status is all about asymmetry. And I'll talk more about that in a minute. And so what we see here is that if the large fish approaches the focal, it's very likely to swim away. But when the focal approaches the larger fish, it's not likely to swim away. We have a much stronger asymmetry in the social roles that they form for the pair-reared animals. Okay, what's happening in the brain? And this is a separate cohort of animals. And we in particular wanted to look at the expression of genes in the brain related to stress and sex steroid hormone signaling. And so again, we had our groups in pairs. We added an isolates group as well. And we kept them in these groups for either 1 week or 5 weeks. And measured a bunch of different genes in whole brain. So the ones in blue are related to the stress axis, so corticotropin releasing factor. Fish actually have 4 receptors that can bind cortisol because of various gene duplication events. So there are 2 splice variants of GR1, glucocorticoid receptor 1, 1A and 1B. There's also glucocorticoid receptor 2, and then the mineralocorticoid receptor. And for the sex steroid hormones, looked at androgen receptor alpha and estrogen receptor alpha. And why did we choose these genes of all of the genes that we could look at? Really well documented across species that they're sensitive to early life. We know that they change over development. We know for this species and others they're involved in regulating behavior. And we also know that they're responsive to behavior. And so that's why we started there. And I want to give a little bit of context for the stress axis. This is a response that happens both to stressors in the environment or potential stressors in the environment. It gets kicked off from corticotropin releasing factor being released from the hypothalamus, signaling to the pituitary to release ACTH, signaling to the adrenal cortex to release glucocorticoids, and both in humans and fish, this is cortisol. For fish, this is actually the interrenal gland, but I'll leave it as it is. And then glucocorticoids can feed back to shut off this axis. And I'll be talking about negative feedback as we move on. And so collectively, this contributes to the stress response. There are also other components as well. And so generally, if we think about a stress response, there's some baseline level of cortisol for an individual, for us as well. When a stressor is detected in the environment, we can ramp up those hormone levels to a particular peak that can vary across individuals. And then the place that we often see variation as a result of early life is in the shutoff mechanism. So once you've hit peak, how fast can you get back to baseline? That negative feedback tends to be really sensitive to different early life experiences. And so MR and GR2 are particularly important for baseline signaling, and GR1 is especially important for negative feedback. Okay, so what did we find across rearing environments, time in treatment, and interaction effects? So we had one gene that responded to treatment effect, which was GR1A, and I'll talk in a minute about why that's so interesting. So we saw in groups, it was significantly higher than in pairs and in isolates. We had two genes, both in stress axis, that were related to duration in the treatment or the age of the fish. So CRF and glucocorticoid receptor 2 tended to decrease in their expression levels in whole brain as the animals went from 1 week to 5 weeks old. Interestingly, our sex steroid hormones signaling through alpha, androgen receptor alpha and estrogen receptor alpha, we saw a really interesting interaction effect where in pairs for both times we saw this significant decrease over time. And then at 5 weeks, we saw that groups were significantly higher than pairs. So something interesting is happening as these fish grow up. And although glucocorticoid receptor 1b and mineralocorticoid receptor did vary across fish, we didn't find that the factors in our experiment predicted that. So it could be something like social status or body size or the sex that these animals are going to become. We don't know yet. When you look at these genes together, because they're not acting in isolation, for pair-reared animals, this is a partial correlation network, we see that there's really strong interconnectivity between the expression of these genes. And in contrast, this is the only significant correlation for group-reared animals. And this is not a mistake for isolates. There were no significant correlations for isolates as well. So I cannot yet tell you that this is the good way to be and that's the bad way to be. We really have to test that rigorously. But something dramatically different is going on for a set of genes that we know are often interacting with one another. Okay, so what have I shown so far? For effects on behavior, we saw that rearing environment shifted behavior in multiple contexts that are relevant to the animal's life. And we saw that we identified this novel behavioral syndrome where the rearing environment shifted where individuals fell along that syndrome. And pair-reared juveniles tended to behave more subordinately, though we don't yet know whether that actually has an influence on, for example, their reproductive success later on. That hasn't yet been tested. In brain, we saw that rearing environment specifically shifted expression of glucocorticoid receptor 1A, which is fascinating because if you look across species, it's GR1 that's mediating a lot of early life effects for animals, where the stress axis function is being shaped early in life, and often it is through GR1. So it was fascinating to me that this is the one that sort of stood out here. We also saw dramatic differences in gene co-expression, where for pairs it was tightly co-regulated, but not for group or isolates. And we also saw changes in just development, so including in stress axis for GR2 and CRF. And so that will be interesting to think about just what's happening along just a developmental background, let alone the kind of environment that you're in. So that's what I was able to show in this experiment. I want to talk now, because it's going to launch the other data that I'm going to talk about today, is what we can't yet say from that. So we had an assumption in this experiment that the juveniles were similar in phenotype at the beginning, right? We took a lot of animals. We mix them together and haphazardly, right, as randomly as possible, put them into these different groups. But we didn't actually measure what their initial social phenotype was, and we didn't actually watch them. And this is what's called the black box of development. And this is really common in early life studies where we say, okay, we're going to put these animals into these environments, we're going to leave them there, and just see them on the other side, without actually pulling apart, okay, what are they doing there? What are the behaviors that they're engaging in? What are the cues through various sensory modalities that are coming in to shape who they become? Another really interesting question to me are, what are the actual social or fitness consequences of this behavioral variation, right? If you look at the folks in this room, I would say we are all very successful, but we don't have the same personality, right? So just showing difference is not yet sufficient to show that there's a consequence for that individual. I'm also really interested in understanding whether the differences in brain that we showed are related to the differences in behavior. And I'm especially interested in what are the functional effects for these various genes on the actual stress axis function. So for example, is GR1A actually mediating real differences in negative feedback? So I'm going to be able to answer some of these questions today. I just started my lab in 2019, so invite me back in a little while, and I'm really hoping to have more on number 4 in a bit. Okay, so the next thing I'm going to talk about really tries to get at number 1, looking in detail at what juveniles are actually doing when they're together to understand what kinds of experiences they're accruing over time, which will interact with their environments, other animals in their environment, to influence, again, who they become. And we hypothesized that in juvenile social groups, similar to adults, that they would engage frequently with each other, and that they would do so within the context of a social hierarchy. We expect that social status will form in all different social groupings of juveniles, and we also expect that larger juveniles, again similar to adults, will have a dominance benefit. So I want to say just a little bit about social status. So hierarchies are super common across species, and it occurs when there's this consistent behavioral asymmetry. So where a subordinate consistently yields to a dominance agonistic behavior. And then a hierarchy will form when there are these statuses between dyads that are organized where more dominant individuals consistently lead to subordinate individuals yielding to them. There are all sorts of different mechanisms that can determine status depending on the animal. And so the reason that I'm going to show you multiple measures of social asymmetry is that it's my goal to understand something appropriate for these juveniles. And even though we know a ton about the adults of this species, there is remarkably little work in the juveniles, even though the labs that study this fish rear them in their laboratories. So here what I did is, with the assistance of then undergraduate, but currently I think almost PhD graduate, Becca Butler, we created these 4 different kinds of social groups. Pairs of fish of the same size, mismatched pairs where there's a larger and smaller, triads where there's a size-matched pair and a larger juvenile, and then mismatched triads of a larger, medium, and small juvenile. And we then measured behavior on the first day they were together, the 3rd day they were together, and the 7th days. Okay, why pairs and triads when we actually care about more naturalistic groups? Start simple. Start simple, we said. We know that nothing in science is ever actually simple, so we'll sort of show you how we've teased this apart. But ultimately we want to try to use this as a basis for understanding large naturalistic groups, even though we know that social dynamics don't scale perfectly with group size. So we know that. I'll give you one context that we already could have a question about. So for example, we saw in the first experiment that within the group-reared animals, there were animals that had a pair-like behavioral phenotype. So that begs the question, are there pair-like environments that happen within larger social groups? So that's one instance of sort of a direct hypothesis that we could build. And why manipulate the size of the fish? Adults are really, really sensitive to differences in body size. There's a whole nother area of science thinking about how animals actually tell their body size in relation to others. I am not going to get into that, but it's not actually known. Like, how do we know how big we are and how do we know how big everyone else is? But fish are sensitive to this, like to 5% of their body weight. So like they're really, really sensitive. And larger adults have a status advantage. It also allows us to socially engineer an instance where determining status might be more challenging for the fish. Potentially not, but we thought we would try. So again, we measured approaches and displacements and calculated multiple measures of asymmetry that I'll talk about. One is directional consistency, another is agonistic efficiency, and the third is a dominant score called David's score. Okay, so I'm going to start with directional consistency, and mostly what you need to know is that if it's a 1, it's perfectly unidirectional. So I interact with you, you interact with the person next to you, they interact with the person next to you, nothing coming the other direction. And if it's a 0, it's perfectly reciprocal across all of the dyads. I interact with you, you interact with me. And what we see is that there's this significant sort of hindrance of the presence of a matched pair. So both for the matched pair and the matched triad, directional consistency is consistently lower than in the mismatched pairs and the mismatched triads. And we actually see the same pattern where the proportion of approaches that lead to a displacement, so the efficiency of those agonistic interactions, is significantly lower in the presence of a matched set of fish. And it's significantly higher when all of the fish there are of different sizes. And so we see this influence on asymmetry simply by manipulating which individuals are making up these social groups. And something about the presence of a size-matched fish is leading to more symmetrical interactions. I would love to talk more about why we think that is. I'm actually not convinced that they can't do it. I think they just might not need to. I have seen fish of the best size matching we could make swim up to each other, stay there for a second, and swim away, and they had figured out their status. Like, I literally was watching and could not, and I missed it, right? So I'm not actually convinced that they can't do it, but I don't have an answer here for what the significance of that is. And then looking at David's score, again, this is sort of a dominance rank that takes into account interactions between a dyad and every other dyad in the social group. And so what we see is that between the animal with the higher or the lower David score, there's a significant difference within matched pairs, not within matched triads, there's a trend, yes within mismatched pairs, and yes within mismatched triads. So we see that statuses differ significantly except for those size-matched triads. And I'm not showing these data here, but large body size was actually only a significant predictor of having the highest David score for the mismatched pairs. There was a trend for mismatched triads and not at all for matched triads. So just to be clear, the larger juvenile is clearly larger. And something about having animals of the same size just in the same tank is completely shifting the social dynamic in a way that I did not expect. I figured that the larger juvenile would easily establish dominance over the others, and that the others would then work a little bit to figure out who was going to be next in line if the hierarchy was going to be linear. And that just did not happen. I'm going to show you one more thing, which is just rates of displacing. So in matched pairs, mismatched pairs, and mismatched triads, the animal that was most dominant displaced the most. That was not the case in the matched triads. Okay, so looking across these different measures, this is just a categorical heat map, sort of looking across for how similar, how weak the status differences were in the lighter colors or how strong the status differences were in the dark colors. We see that these different social environments really had a big effect, bigger than I thought. So we can clearly say that juveniles can form status relationships. That's not super surprising, but this is the first in this species to actually quantify these different metrics of asymmetry. Something about having size-matched fish present decreases the asymmetry, which, again, coming back to our bigger picture, seemingly to us, subtle variation in the makeup of the social group potentially leads to really dramatic differences in social dynamics. And so again, if you're thinking about baby fish who are having their first experiences out in the world, the kinds of experiences that they're accruing in different kinds of social environments are going to start to be quite different as they accumulate. And so in the final two studies that I want to tell you about, we basically tried to take advantage of what we found here. First in the matched triads, basically saying, can we take a social dynamic that was really quite symmetrical, and can we use pharmacology, manipulating in the brain, to promote social asymmetry? And then using the matched pairs, where they were strongly asymmetrical, can we use that to identify what the mechanisms regulating status are? Using a really strong dominant-subordinate phenotype for juveniles, can we go in and say, what's happening in the brain, what's happening with hormones? And here we're going to compare that to what's happening in adults to test whether the regulation of social status is maintained over life history. So in thinking about the neural mechanisms regulating social behavior and status, I will say that there are many. In this study, again with Becca Butler, we chose to focus on the nonapeptide vasopressin, and we hypothesized that it affects social behavior, status, and the expression of genes involved in stress, sex, and nonapeptide hormone signaling. And the reason that we chose vasopressin of all of the sort of usual suspects that you could look at in the brain for a couple reasons. One, if you're comparing, has a highly conserved role regulating social behavior and status. Some of the most famous work is with pair bonding in voles, or social grouping, sort of pro-social behavior, both social approach and are you willing to aggregate in large groups, or do you prefer to either be by yourself or with one, let's say, mated pair? We have a place to start. Vasopressin has been studied in the adults of this species. Receptors have been mapped. Location of the neuron populations in the preoptic area of the hypothalamus have been mapped. So we know quite a bit about it. And vasopressin has a role initiating the stress axis. So we have this connection to something that we think is happening in early life. And our prediction that injecting with juveniles with vasopressin will make them more aggressive and more likely to be dominant. So in this study, we used these matched triads. We had a larger juvenile that was uninjected. And then for the size-matched fish, we injected one with just a vehicle. And we injected the other either with an agonist or an antagonist. Basically saying, okay, larger fish, here are two fish that look the same. We know they don't, right? No two individuals are exactly the same, but, right? And we're shifting one by up or down regulating the signaling of this system. Just to give an appreciation of Becca's work here, if you've seen the cap of a microcentrifuge tube, this is the size of the juvenile brain. And you can see this little blue dye here. So this is an injection that has hit the third ventricle. So basically in this operation, I anesthetized the fish, I tagged them, handed them to Becca, Becca injected them, and they were back in the water in 2 minutes. So this was like quite the operation. Okay, so what did we find? So first looking at David's score, this is the same dominance index that I showed you before. You will very clearly see that the larger juvenile is dominant over everyone else. And whether you're in the vasopressin groups or the Manning compound groups, which is this antagonist, there's no significant difference in status. They are the same status. And I'm not showing you this here. I have the data in the after slides if we want to look at it. It also did not have a direct effect on behavior. So no effect on focal fish behavior, no effect on status. What we did find really interesting is that the larger fish directed its approaches, it directed its social attention to one fish over the other. And it did so to the fish that presumably had the lower vasopressin signaling. So in the vasopressin group, we're elevating vasopressin in the drug animals, so the control animals should have lower vasopressin, so they received more of the attention, and it's the opposite for the antagonist group. So something is going on that we are not detecting the mechanism by which it's affecting the social behavior of someone else in the group. We also did a principal components analysis across the gene expression and behavior and status-related behaviors. We saw that for principal component 2, which is really related to vasopressin manipulation and signaling, that PC2 values were significantly higher for both the drug and control in the vasopressin group, significantly lower in the Manning compound, suggesting that this is an effect not from the injection, because the control animal didn't get the drug. It's something happening as a result of the shared social environment that's leading them to have more similar signaling in the brain. I will also point out that we have in PC1 this suite of genes and behaviors that are representing social status. So here we see that David's score, our dominance index, is positively correlated with glucocorticoid receptor 1A and 1B expression. And in the opposite direction, the tendency to submit is correlated with mineralocorticoid receptor and glucocorticoid receptor 2. So here we show that there was no direct effect on behavior, so against our prediction, and it did not significantly shift status, but it did affect behavior of the non-injected fish. And we suggest that this is because that fish was more likely to become dominant in the future, and we have a couple of reasons to think that. We don't yet know what the cues for the larger fish are, but I will note that there are studies actually in anoles and in rhesus macaques that showed the exact same thing, and they don't know either, where they manipulated levels of vasopressin or oxytocin signaling, which are really closely related, and they share each other's receptors. They saw an effect in the non-manipulated animal, and they also could not detect what was changing about that animal. So we are in good company, and presumably we will know more soon, right? There's got to be some cue, whether it's chemosensory, whether it's a subtle behavior, right? There are a lot of possibilities there. And it was super exciting to identify this integrated brain and behavior status phenotype, where high status was associated with GR1A and 1B, low status associated with GR2 and MR. Okay, the last thing I want to share with you, and then I will wrap up, is ongoing work in my lab. I started my lab in 2019, so I had about a year before the pandemic hit. This is a really exciting study, and I actually think it's sort of the best of working at a liberal arts school. So this was done in collaboration with my colleague, Dr. Finley Finseth, and the experiment was actually designed by her undergrad bioinformatics class. So I came in, I presented for about 10 minutes about the fish, suggested some elements of experimental design, then the students went off into breakout rooms and made some sketches, came back, pitched different ideas, and then I went in and ran the experiment, gave the brains to Finley. Finley did the RNA-seq on them, and then my thesis students took over. Christy did the size measurements, and Jess and Bella did the hormone measurements and the behavior measurements. So really, really cool example of collaboration sort of across all areas of what we do. So, and what we hypothesized is that across juveniles and adults, dominants and subordinates, that there would be similar mechanisms of social status across the behavioral, hormonal, and neural levels. So here we're taking advantage of that mismatched pair dynamic. So we had a larger and a smaller juvenile, a larger adult male and a smaller adult male, and we did include 2 smaller females to create sort of a reproductive context. The males, if left alone as adults, they will get too aggressive to stay there by themselves. So adding the females in creates some more naturalistic social environment. And we kept them in these groups for 1 day or 7 days, and then took measurements across behavior, hormones, and brain. We looked at waterborne hormones for cortisol and testosterone, And then we did whole brain transcriptome analysis using TAG-seq. Okay, so what did we find? These are all going to be measures that you've seen before. All of these different groups, the juveniles after 1 day, adults after 1 day, juveniles after 1 week, and adults after 1 week, had significant differences in this social dominance measure. For directional consistency, we actually saw, for this and for agonistic efficiency, we saw a significant effect of time. So something about the social dynamic takes some time to establish, which is not super surprising. So we see that directional consistency and agonistic efficiency are higher in both weak and adult, weak juveniles and weak adults. Now you'll notice that the adults have a lot of variation here and here, so it's kind of interesting. When we're looking at cortisol on the left and testosterone on the right, we saw two different patterns. We saw in both the juveniles and the adults, cortisol was significantly higher in the dominant animals, but it took a week to establish that pattern. This is not super surprising. There are studies in the adults of this species that show dominants have more cortisol, some show subordinates have more cortisol, and some show no difference. It really is sort of context dependent. And for testosterone, the adults only showed this pattern, not the juveniles. They had significantly higher levels of T, and the dominants had significantly higher levels than subordinates, which is perfectly consistent with what's been shown before. And in the brain, we see this dramatic effect of developmental status. So this is a principal components analysis, PC1 here, PC3 here, and we see this perfect separation of adults and juveniles. And you can also see this time effect where day is up here and week is down here, and some but very few differences related to status. And the scale of this differential gene expression is dramatic. So when we're talking about juveniles versus adults, we're talking about hundreds to thousands of genes are differentially expressed. When we're talking about day to week, we're talking about dozens of genes. And we're talking for DOMs and subs, it's like between 0 and 2. So something really, really interesting is happening here across our different factors. So as I'm wrapping up, just a couple highlights. In looking at the weak juveniles, vasopressin is significantly related to status. For those of you who have any experience with sequencing of this kind, the candidate gene you are interested in never pops out. And here we have the exact gene that we were interested in, showing as this really important difference for juveniles, but not for adults. And in adults, we have some really interesting, especially neuroendocrine genes related to status. But really what we're seeing here is a developmental signal. Okay, so juveniles and adults clearly had status with some differences in their asymmetry patterns. Status differences for cortisol were similar for juveniles or in adults, but different for testosterone. And we saw the largest brain differences in this developmental signal. And moving forward, what we want to do is not just analyze each level separately, but actually map across levels. And finding these points of similarity or difference are going to be really strong in making predictions about what's happening at the other levels. Okay, so some key takeaways. I hope I've convinced you that there's some critical developments in behavior during early life, and they may have this really important lasting impact on phenotype. Behavioral and neural phenotype is shaped by multiple kinds of probably interacting early life effects. I really only talked about social effects, but there are maternal effects as well and others. Social sensory cues and social experiences may be key to that behavioral development, and it can create those behavioral changes by changing the neural and neuroendocrine mechanisms underlying them. And in particular, my work, I hope, has shown that stress axis and vasopressin, as well as some sex steroid hormones, may serve in this conserved role, mediating the effects of early life environments on brain and behavior. So we saw again that there was this GR1 effect from early life social environment, perhaps influencing negative feedback. So if you have higher expression in the brain, especially in hippocampus, it can make that return to baseline happen quicker. We saw that higher status was associated with elevated GR1A and B, decreased GR2 and MR, and then increased cortisol. We saw influences of vasopressin in terms of competitiveness, attentions from others, and juvenile social status, and we know that vasopressin can kick off the stress response. And we also saw changes over time in CRF and GR2, so thinking about these developmental changes will also be really useful. And then we also have exciting ongoing work basically in all of these areas coming out in my lab as well. And so with that, thank you all for listening. I'd like to thank everyone who contributed, and I'd be happy to chat.
Why don't you take questions from the room first, and then we'll...
Okay, yeah, I see one on the meeting chat as well.
Oh, okay.
Well, that one's fast, so I can answer that. So the possible effect of returning faster to baseline. So prolonged exposure to glucocorticoids can have really serious negative effects across different levels of physiology. And so if you shut off a stress response faster after it initiates, you're basically cutting the amount of time that the body is exposed to those high levels of hormones. Yeah, please.
That does seem to be pretty crucial in whether a stressor is like, or a stress response is adaptive or maladaptive.
Yeah.
And you said there was a lot of variation in that different baseline, and I didn't quite get it. Were you able to pick apart what was explaining that variation at all?
So I can tell you from other species that the location and density of glucocorticoid receptors, especially GR1 and especially in hippocampus, are really important for shutting it down quickly. So we have a lot of examples actually through rodents where this has been studied in detail, where you can actually track, let's say, different maternal parental environments having a direct influence on, through epigenetics, on the number and density of glucocorticoid receptors in hippocampus, having a direct consequence for that negative feedback. And so we have not mapped in the juveniles what the degree of variation is in GR1. We actually don't have antibodies for GR1 that work in fish, so we're going to have to use some other methods. But given how conserved that is, that's a really strong hypothesis to test. But I haven't gotten there yet, but it's sort of top of list given everything we've now shown that really gives us some clear directional hypotheses about who should have what kind, should have what kind of stress negative feedback, and then we get to test that.
I'm really curious about that stage where they're in their mother's mouth. And
Yeah.
I'm guessing maybe I missed this, but if you control for that, so when you make your pairs and triads where they all have the same mom from different moms, and then can you manipulate like how many fish the mom has in her mouth?
Yeah, that's a great question. We have not done a ton of work there, but it is absolutely feasible. So for these experiments, we had, I think, 6 to 8 mothers contributing, and then took the approach of, rather than trying to control every single part, we put all of the babies in a bucket, sort of let them do their thing for a while, and then tried to haphazardly assign from there. We did a small study in Hans's lab when I was there, taking babies out early and then just rearing them in a tank, and then leaving another set in from different mothers, so that is an issue. The ones that came out early, I believe, were not as quick to socially initiate as the ones that stayed in mom's mouth for the full development period. But by a week later, they had made up that difference. And one of the challenges in understanding early life effects is you can sort of build a just so story either direction, where it's like, okay, these babies have been out longer, they've got more street smarts, they should, be better at it. Or they didn't get what they needed early on, so they should be worse at it. And so you really have to think carefully about how you design these experiments and these hypotheses so that you're not just fulfilling whatever you'd like. But we do have some inkling that that early manipulation does matter. We can take eggs out of mom's mouth and put them back in, and most will keep them. Sometimes they will burst as you're moving them. Presumably we could cross foster. Eventually that would, if we were looking at maternal effects, that's really what we would need to do. But we haven't tried that beyond just sort of trying it. Not in a full experiment, but it's a great question. Yeah. Yeah, please.
So when a dominant male is chasing subordinate males, how does it do, like, not to chase females away? Because at least in the tanks, it looks like there's a negative effect in that sense, right? And also thinking along that line, like, how does the juvenile interaction may affect that? Like, is it only males, females?
Yeah, two really, really good, or multiple really good questions. So I'll take the last ones first. We actually cannot sex the juveniles non-invasively, which is an issue. They do have a sex ratio of about 1:1, and we've allowed cohorts to grow up and actually measured that, and it is in our hands about 1:1. So for the babies, everyone is interacting with everyone. And I don't have a sense yet of what influence that has. I do have a data set where we let animals grow up, and then from the sex ratio, from identifying their sex once they were mature, went back to their early stages to try to figure out were there sex differences. There were some subtle sex differences earlier than I expected, but it was not especially dramatic. And animals tended to be pretty plastic. They weren't really repeatable in their behavior over time. So that needs to be tested again now that we have some hypotheses there, but we can do it post hoc, but we can't currently do it in the moment, which is an issue. For the males, what's really interesting is they're pretty directed with their approaching. So in the video that I showed really early on, here, there's a whole group of animals up here, and he can actually approach one fish, and that fish will swim away, but the rest of the group will just stay put. And so for a female, he can do a courtship behavior at her, and to a fish right next to her, do sort of an aggressive interaction, and they seem to have no problem differentiating between what that is. There is some chasing that's involved in the courtship. The male will sort of circle back and bring the female to sort of chase her over and lead her to where his territory is. And one of the things from the last experiment that I didn't show you yet is what's happening between the focal males and the females that were in the tank. So it's very possible that the adult groups aren't as asymmetrical because they have this other reproductive context. So that very well could explain what's happening there.
We have a question in the chat.
Oh, yeah.
You can also try to unmute the monitor if she wants to.
Oh yeah, there are a bunch here. Okay, curious about the stage where they're in their mother's mouth, if you controlled for that.
No, that's me.
Oh, sorry.
Go to Molly Fox.
Oh, got it, okay. Thinking about the stability of social hierarchies, particularly in juveniles, and what this means for how juveniles interpret cues. Because assuming juvenile growth is not 100% linear, which individuals are larger than others may be constantly changing. So I wonder about the tracking of social hierarchies during these early stages of life. What are juveniles gaining adaptively? Yeah, really, really good question. And I will try to get to some of that. So you are absolutely right that juvenile growth is not linear. We've actually tracked in that by 2 months old, a shared community of, let's say, 150 fish, the largest juvenile is like 22 times bigger than the smallest juvenile. But the small animals are not necessarily bad growers, because then if you split that community out into same-sized groups, smaller groups, even the tiny fish will get these really accelerated growers among them. And we think that has to do with like social suppression to some degree, and I'm sure there are some growth components to that as well. in terms of tracking how that's going to influence the hierarchy, so one of the things that we see in adults that we don't know if we see in juveniles, adult males expend a ton of energy, right? They are total busybodies, and they have to defend their territory actively, even though it's a stable group. And so one of the things that we see in the subordinate males is that they invest in growth. They're not investing in reproduction, they're actually reproductively suppressed, and so they can then grow to a size at some point they can challenge and then ascend to a dominant status. And so because juveniles are sort of in the business of just getting bigger, I don't know how status lays on top of growth trajectories, and it's a really fascinating question. We do have a study in the lab looking at growth patterns for animals of different sizes in groups of same-sized animals, and looking at growth trajectories and how testosterone and estrogen change over time. But that was during the pandemic, and we don't have behavior from that. So that's really what we'd need to sort of figure that out. But you're absolutely right. I would expect there to be a dynamic, nonlinear interaction between maybe initial size, growth, initial status or position, and then interacting with what future, behaviors are, sort of thinking more about this figure here where everything sort of feeds forward. But I don't expect any part of that to be linear.
I have a question if no one else does.
Yeah.
I just love this system. I work with monkeys and they're such a pain, and we try to do a lot of this stuff, and it's just much more difficult to move them and manipulate them. But you're saying that you can't tell the sexes apart. It's a problem we have too. And one thing we found is one of our best cues of what sex they turn out to be is how the other monkeys treat them. Mm-hmm. So for example, in our system, all males are very, very interested in alloparenting male babies, but not females.
Oh,
and then the females are interested in both. I was just wondering if there are chemical signals and non-visual signals that the fish can tell before you can tell, and whether that would be something you could look at later. I mean, it's kind of cool that you don't know because it's a blind experiment.
True.
You're coding, you don't have any idea what sex they are.
Yeah.
So.
They absolutely use chemicals to communicate, and there's a lab that also studies this fish, Karen Maraska's lab at Louisiana State, that does a great job studying chemoreception and how the brain responds to different chemical cues, and there's some really interesting stuff there. We have not done anything with that, but I have no doubt that they know before us. I assume that if there's something that we can see, for the animals, that cue is just like so extreme. One thing we do see, which is curious to me, so you saw for the adult males, they have this really prominent eye bar, the black eye bar. One of the things that we see for juveniles, and it does not seem to be related to their size, to their relative size at the time, they will develop a black bar just through sort of the eyeball portion. And I don't know whether that happens for everybody at some point, whether it just happens for males at some point. Females, if you're put them in an all-female group of adults, they take on dominance characteristics, including the eye bar, so it's not that females can't do it. But that seems to be some visual signal that we could sort of tease apart what that is. I'm sure they know. I'm sure they know. We also have an experiment that we just got data on from sort of different points in development, including babies right out of mom's mouth, and hormones like testosterone and estrogen are like extraordinarily higher than I would have expected. So I had a colleague that basically expected us to need to grind up the fish to be able to get sufficient levels of hormones, and we can actually get it from the water. So if you put a baby fish or any fish into a beaker of water, especially steroid hormones, because steroid hormones can cross membranes, they will excrete through the gills and through urine and feces, and so you can actually extract hormones non-invasively through the water. And so we were able to do that for day 0 fish, which was shocking. And, you know, for folks who sort of know about organization activation, for mammals at least, right, you have a quiescent period for the gonads post-development before, you know, until puberty starts. Fish do not follow those rules, but I also never dreamed that they would have measurable levels. So if we can measure them in that way, like, I am sure they're literally just swimming in it, but I don't know what those cues are. It's a great question. Yeah. Yeah, please.
Species, is there like a stark difference in reproductive success between like dominant males and subordinate males? So like, is it more adaptive to be dominant?
Yeah, it's a really good question. So only the dominants are reproducing.
Okay.
And so to understand reproductive success, you would need to do sort of an additive over an animal's lifetime. What's the period of time that he's investing in reproduction? What's the period of time maybe as a subordinate that he's investing in growth? So that really classic life history trade-off, right, of growth and reproduction. Because you could have an animal that is optimally switching, right? Because you wouldn't expect the highest reproductive success be to the male who just stays dominant forever. Maybe in the lab, it's a pretty cushy life. But you wouldn't expect that in the wild, right? Energetically, it is just so costly what they're doing. And so optimally, you should see a particular pattern of switching. I described the adults as dominants and subordinates. There actually are some intermediates that aren't fully suppressed. I don't think I added the video here, but Isaac, the same scientist who got the group videos that I showed you, got this video of a male courting a female, and the shoal is up here of subordinate males and females. And then right as she lays her eggs, this intermediate male swoops down, releases a bunch of sperm, and gets back out. And so there are reproductive events that happen in that way. It's probably not cumulatively as successful as having your own territory, but there's a whole area of research too on alternative male reproductive strategies. So it's not quite as stark as I described it. But yeah, it should be a particular pattern of switching. Yeah. Yeah.
So in your DC analysis, different times, did you see any of the genes you wanted to look at a priori in the list? Because you had 3 different measurements. Were any of those in the 2 categories above the one that had vasopressin and
It's a good
and
question.
HIF? And then also, what do you think is happening with HIF?
Let's go to there. Probably my answer is I don't know because those data are brand new. And this is the first time I am presenting them. So thinking about hypoxia-inducible factor, I don't know. It's a really good question. Do you have a background knowing about that one?
About HIF?
Yeah.
I do, and I was just wondering if like oxygen processing could lead to higher sizes.
I think
Larger
Yeah.
body size
it's very possible, and one of the reasons that I'm so
or energy.
Right. One of the reasons I'm so interested in stress axis is I sort of didn't include it in the summary here, but like all of this is functioning to mediate growth and metabolism,
Right.
right? And so when we're talking about baseline signaling through mineralocorticoid receptor and glucocorticoid receptor 2 and thinking about cortisol, like you need that to grow, right, and process energy. And so for juveniles, if we have evidence that it's playing this role in this social early life effects world, it is also doing these other things, right? And so I think that's a really good hypothesis. I have not looked into it at all.
And it's only 2.
I know. I mean, it was shocking. I mean, it's also not shocking, right? Because really similar brains for animals like this need to be plastic, right? They need to be able to do whatever their environment requires of them, and so you're not going to need massive reorganization to sort of switch if you're switching naturally over time. That all being said, I'm not sure I would have anticipated this. I was expecting the biggest signal would be juvenile and adult, but not to this degree. But yeah, it's a great question, and that makes a lot of sense, and it makes a lot of sense in terms of thinking about the other axes that are involved. But I don't know. Yeah.
No more in the chat.
Okay.
Oxygen deprived acidified environments reduce growth.
Yeah.
So,
And that could be also something that we manipulate, right? Because in a lab environment, you could change dissolve and measure dissolved oxygen. Yeah, food restriction or just, changing the amount of food. We discovered that there are tiny, tiny plastic spoons that you can buy on Amazon for like making your own pills, but they're actually perfect for feeding tanks of fish the exact same amount without actually having to weigh out the amount of food each time. So like we tried to control for, amount of food per number of animals, but you're also not able to watch like, okay, is everyone getting, we think everyone's getting some. No one is starving, they are fat and happy, but like, are they eating equally? Like, probably not. Yeah.
Stress responsivity and GR1, and I'm wondering if you thought about an experiment where fish are exposed to acute stress because you're able to measure cortisol in the water. Enabling you, I guess, to look at stress responsivity in adulthood or plasticity throughout development. Is that something that you've thought about doing?
Yeah, it's a great question. So we started at the end of the summer trying to work that out. One of the great things about the non-invasive measure is that it's non-invasive and you can do it multiple times. One of the challenges is it's really giving you a sum of hormone over a time period rather than like a really fine scaled time course. Did you have a?
Excreted through the gills, and if it's probably really stressful and the fish change the water. So you could do it.
Yeah, so what we did this summer, and we'll see what the results are that will give us a baseline basically to jump off from, is had a bunch of really young fish. We wanted to see if we could do this as young as possible, so we used fish that were less than a week old. We actually made our own nets so that they like wouldn't get lost in them because that can be a stressor. So like the student that ran this project, Allie, like constructed her own nets and they are adorable. And so she had a group that were just in a single beaker for 90 minutes, a group that we changed beakers 30 minutes, 30 minutes, 30 minutes to see if we could try to get a rise, peak, and fall, but it could also just be a new spike every time you move them. I'm not exactly sure. And then we included two additional sort of common lab stressors. One was, it's not really confinement in a net. The size fish we're talking about, like they're like, 6 millimeters long, but we put them into a beaker where a net was lining it. And so it's sort of a different environment. So we put them there for 4 minutes and then did beaker, 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 30 minutes, 30 minutes, 30 minutes. And then we did one where the first 4 minutes they're on an orbital shaker to just create some movement. Our pilot data actually suggests that that was more calming, so we'll have to see. But again, just trying to work out some of the methodology of what range of cortisol are we even talking about for animals of that age. Pilot data suggests it is very high. Not exactly sure why, except other than the conversation we just had of like their job right now is to like grow and develop. But we don't yet have data on whether the transfer works. There is a system of being able to basically drain water into a catchment and then sort of refill at a particular rate so that you're not getting significant mixing. We don't have that worked out yet, but it's possible. But yeah, we are absolutely wanting to go there to figure this out. We can't take blood when they're that little, and so it has to be this other way. Otherwise, we'll never get at that. Yeah.
I was just amazed at the amounts of hormones that are in the water secreted by them. So can that be taken up by other fish? Because it's causing a really striking hormonal environment, just chemical environment that affects the further production of these hormones in the other fish that are swimming in someone else's cortisol excretions.
Yeah, it's a great question. So yes, they do excrete them at high levels, and that correlates with what's in plasma. So there have been a lot of studies sort of taking a blood draw and taking waterborne hormones and seeing that positive correlation. Whether the hormones or what role excreted hormones take in manipulating the physiology of other fish. I don't know a ton about. There have been, this isn't quite that answer, there have been some social buffering studies, similar to the ones that have been done in like prairie voles and meadow voles, of if you take animals that have been co-housed, or for the voles that are pair bonded, you take one out, you stress it out, and then you put it back with either their familiar animal or a non-familiar animal. The social buffering can lead, being back with the familiar animal can again lead stress to sort of shut down more quickly, so that over time you have sort of less exposure to glucocorticoids. And so there we're seeing a direct effect of the sociality on that animal's hormones rather than the excreted hormones stressing the other animal out. But they've got to be active to some capacity, and it's a fascinating question, and I don't know the answer. Yeah.
We don't know what kind of olfactory signals they get, but we do know that they're super interested in other animals' pee. For example, if males see a female pee, they run over and elbow each other out of the way to get access to that urine, and they sniff it and look and think about it. And we don't know what's in there, but it just seems to me that there must be so much more of that. Maybe not as directed information, but being next to a fish who's secreting stuff at a high rate, presumably they have some directionality, even though they're picking up on the stress levels of their neighbors.
Yeah, and there's really beautiful work again out of Karen Ruska's lab of exposing dominant males, subordinate males, and females to urine from a female that is gravid, so ready to lay eggs, and you can see really dramatic differences in olfactory neuron activity and other plants in the brain in response to that. And so if you have a male, basically the brain changes, right? So if a male can be dominant or subordinate, subordinate males do not respond to the gravid female's urine in that way, only the dominant males do. Dominant males don't respond to the urine of other dominant males, but subordinate males really have a strong response to that, because that's sort of their main focus in the tank. And so they definitely are using it for communication. Yeah, like undoubtedly. But it's fascinating to think. And all of this, of course, is like stress within context, right? So like, I really like to reject the idea that being dominant is good and subordinate is good and dominant is bad, because over time these animals are going to fill different roles with each other, even simultaneously. They're going to, if they're males, they're going to occupy dominant and subordinate status potentially over their lifetimes, and so it's really about how they fill that role rather than can you avoid sort of quote unquote the bad one. And their interactions really are, even for the juveniles that were really tussling, the vast majority of the time they're not hurting each other, right? It's really pretty stereotyped. And so, they've evolved to be in that context and sort of thinking about how they fill that and thinking about how stress axis mediates some of that. But again, we're not talking about sort of miserable animals that aren't growing or eating or any of those things. And so it's an interesting thing to sort of hold in both hands, because it does have an effect, right? Undoubtedly it has an effect, but also. So.
We have time for one more question. Is there anything else? Renee, last question.
It's more of a clarifying question, actually, I think. And it was about this slide. I was just wondering, and sorry if you had explained before, I didn't really catch it. Like, what do you think the dimensions in this principal component analysis, what do you think these different factors represent?
Yeah, it's a really good question, and like I said, I probably did not explain this well, because these data are brand new. My collaborator sent these to me yesterday. We're both like super excited to share this with someone. It looks like dimension 1, which is 16.5% of the data, is really giving us this developmental stage information. So independent of week or day, independent of dominant or subordinate, it's really giving us a signal of are you a juvenile or are you an adult? Again, I guess I would have expected that to be a really strong signal. But given the fact that they're doing a lot of the same things, of course it happens in a completely different context. For the adults, it's within a reproductive context. For the juveniles, it's not. But basically what this is screaming at us is that if you look at whole brain gene expression, the major differences that are coming out are you an adult or are you a juvenile? And then for dimension 3, this basically gives us the best, looking at 1 versus 3 gives us the best separation of the factors that we were interested in, is giving us this, were you together with your social pair for a day or were you together for a week? That was actually especially exciting because that was the addition that the students in the class were like really dead set on. They really wanted a time component to understand how social relationships develop. And so we sort of settled on these 2 snapshots of, okay, you've been together for 24 hours, you've been together for a week. And so that's really what dimension 3 is pulling apart. And so up here we have the day dominants and the day subordinates for adults. Here we have the day subs and the day doms for juveniles, and then down here we have the ones that have been together for a week. So what's happening with the hundreds to thousands of genes that are represented in there, especially between the juvenile and adult. We do have some, and my thesis students have done a bit of this, pulling out some candidate genes, but really what we want to do is look at it at a much more holistic, integrative level, thinking about behavioral phenotype in these multiple dimensions, thinking about the hormones that we have, and then thinking about gene expression. Because we actually have quite a bit of detail for the approaches and displacements. We didn't just do one version of approach and one version of displacement. We actually categorized based on the distance that the animals started. So were you close together when the approach happened, and then did that lead to what kind of displacement? How far away did they go? Or did you like swim from across the tank to like, get going? My students talk about this as the best friend hypothesis. So you might interact with your friends when you're sitting right next to them, but like they're probably, as a response to that, not just gonna like run off. Whereas if someone from across the room came sprinting at you, like, yeah, I'm gonna get out of Dodge, right? And we can analyze this in sort of a different way than I've mentioned here. We can look at sort of, okay, what kind of approach was it, and then how far away did the other animal move? So there's a layer of complexity here that I sort of wasn't able to talk about here today. But thinking about, okay, if we have juveniles that approach from a distance, but then they don't actually move very far, what does that mean in terms of status, right? If I walk up to you and you're just like, oh, hey, rather than running away, that means something different. And so thinking about, again, sort of finding points of similarity and finding points of differences to try to pin down what kinds of expectations we might have for hormones in the brain. So that's the direction that I'm really excited in going.
All right, I think the formal part is over.
Okay.
We want to let you eat lunch, and if people have a few more questions, maybe they can chat informally during the next half hour. Otherwise, thank you so much. It was
Thank you
fascinating.
for having
so much
me.
information.